Okay, so the major significance of this specific study was that it was the mark of the first major outbreak in a Delta form of COVID, despite the majority of said population previously being vaccinated. The particular outbreak in this study, quote, raised public health concerns and attracted international attention, end quote. Furthermore, discovering the transmission rates among the Delta variant infected individuals who had received vaccinations as well as individuals who did not in Provincetown and in the country can prove highly beneficial to the researchers. These findings can help lead to better understandings of variants as well as additional solutions to eliminate its spread.
This study took more of a discovery-based approach rather than testing a direct hypothesis. However, a valid hypothesis implied from reading the paper is, if viral genomic data can be identified, then modes of transmission among vaccinated individuals can be better identified. This hypothesis is derived from the phenomenon that vaccinated individuals were spreading this novel form of the SARS-CoV-2 virus identified as the Delta variant. Scientists were able to test this hypothesis accurately and efficiently due to a, quote, robust public health response, end quote, that, quote, permitted extensive epidemiological and genomic characterization of the outbreak and the structure of transmission within it, end quote. Using modern gene sequencing technology and diligent contact tracing, the team of scientists conducting the study were able to understand how quickly the Delta variant spread among vaccinated individuals, along with how it was/is spreading. By testing this hypothesis, the team now has, quote, generalizable insights for containing future outbreaks of Delta and other highly transmissible lineages of SARS-CoV-2, end quote. The aim of this study was to track the transmission of the SARS-CoV-2 Delta variant amongst vaccinated individuals. This data is important in order to have significantly better public health responses when there is a large outbreak of a variant in a densely populated town. Some key things the scientists in the study wanted to test was the, quote, epidemiological and genomic characterization of the outbreak, the structure of transmission within it, and the role of vaccinated individuals, end quote. All of this for, quote, containing future outbreaks of Delta and other highly transmissible lineages of SARS-CoV-2.
The data used for this study was obtained by the Massachusetts Department of Public Health mobile testing stations. Once gathered, it was sent to Massachusetts State Public Health Laboratory, or the Broad Institute, for sequencing. The data was then analyzed for disparities. Differences in age and sex studied ranged from 0 to 105 years old, as well as 94 females and 373 males. The data included both vaccinated and unvaccinated individuals and assigned indexes for risk of transmission between these individuals. Finally, a phylogenetic tree of the sequenced Delta genomes was constructed. This allowed for the transversion of the tree from the individual tips to the earliest common ancestor, an accurate proxy for a transmission timeline among individuals. Finally, they compared nucleotide substitution frequencies of the Delta variant in Massachusetts to the Delta variant across other areas of North America to compare the relatedness of each Delta outbreak. This was done to determine the origin of the Delta outbreak in the United States.
In the 467 cases of COVID found, there were no alpha variants in the Massachusetts population. 99% of the contracted cases were carried by the Delta variant, whilst the other 1% was carried by Gamma. It was also found that 83% of all of those Delta cases came from a single source. This indicates that it was a super spreader event. There was one person transmitting to many, many different people. However, despite the signature of a super spreader event, it was found that this was not true nor feasible. That, in fact, there were many different interactions from many different people. The common lineages is attributed to a low mutation rate within the Massachusetts Delta population. The intra-host single nucleotide variants inside of the Delta virus had no significant difference between vaccinated and unvaccinated individuals, which means that the Delta variant used the same machinery for vaccinated and unvaccinated individuals. No immunity or resistance was conferred from those who have been vaccinated previously against alpha. Finally, we know that this lineage of Delta that came from Provincetown was largely confined to Massachusetts. There were a couple of out-of-state cases, but generally speaking, it was all confined to Massachusetts. This was contrary to what the researchers had initially conceived when they were conducting the research. They had thought that the Provincetown Delta variant was responsible for the majority of cases, or at least a reasonable percentage of them, in the United States, that it had spread outside of Massachusetts and succeeded beyond there. This was found not to be true. It turns out that there were many introductions of the Delta variant throughout the United States before and after Provincetown. Provincetown was only notable because of the breakthrough infection rate. Because almost everyone there, 84% of the population, was vaccinated. It was surprising to see that much breakthrough. So why were the morbidity and mortality rates in Provincetown and Massachusetts in general so low compared to the rest of the United States? The authors actually attribute this to the vaccination. So we know that Delta is more lethal than Alpha in unvaccinated populations, but because of Massachusetts' high vaccination rate, the symptoms of Delta were very mild, despite the fact that it has moderate to high viral loads, and definitely higher than Alpha. This is contrary to what was found with the intrahost single nucleotide variants, because they were similar between vaccinated and unvaccinated individuals, but somehow vaccinated individuals were conferred some kind of resistance outside of that. The mechanism of action for that is unknown. The authors attribute the localization of the outbreak to be primarily due to the public health response of Massachusetts, and that includes indoor masking mandates, mobile testing, extensive PR, and high rates of vaccination. But they also note that another big factor may have been the historical public health cultures of Massachusetts, specifically the Massachusetts gay community. Many of those individuals lived through the 1980s GRID epidemic. Before AIDS and HIV were known as such, it was termed GRID, gay-related immune deficiency. And those who were aware and took measures against GRID were selected for in a way. That zeitgeist and culture still persists, and it was passed possibly into the wider public within Massachusetts, which kind of indicates that the biggest prevention measure against any epidemic is culture and education, how people interact with the epidemic and what they're willing to do.
Individuals vaccinated for SARS-CoV alpha did not gain immunity to the SARS-CoV Delta variant. Researchers determined that the July 4th outbreak in Massachusetts was due to the Delta variant being transmitted independently multiple times. Despite signatures of one super spreader event, the majority of the infections were found to be transmitted from vaccinated hosts to vaccinated individuals. The rapid decline and tight localization of the outbreak suggests that the Delta-driven outbreaks are not curbed by high vaccination rates. They can be inhibited by educating the population about safe public health techniques.
The results of the study indicate that there are other methods to prevent future outbreaks of the SARS-CoV-2 variant, along with other pathogens beyond the development of the vaccine. These measures include mask mandates, social distancing, and other simple public health measures to minimize transmission and contraction of pathogens like the Delta variant. In the future, the information obtained in the study can help communities limit pandemic-level threats from deadly pathogens.